We're very happy today to have Henrick Hill, MD, PhD with us. He's a pediatric endocrinologist focused especially on the management of type 1 diabetes at Uppsala University Hospital in Sweden. And in addition to his vast clinical experience, he's currently preparing to defend his PhD thesis entitled Investigations of Hypoglycemic Events and the Role of GABA in Type 1 Diabetes. His research includes analysis of endogenous GABA levels in individuals with type 1 diabetes, as well as the results from a clinical intervention trial in which the potential beta cell regenerative capacity of GABA in patients with long-standing T1D is investigated. The thesis also explores the occurrence and frequency of hypoglycemic events in children and adolescents. I will drop two of the papers into the chat, and you can drop questions in the chat during the presentation. He'll take some questions at the end as well. Thank you, Henrik, for joining us. Really appreciate this time and really curious to have you walk through the data with the scientific audience.
Thank you for having me, Monica. As you know, I'm a fan of the sugar science, so it's an honor to be invited.
Thank you.
I actually can title myself a PhD now. When we wrote this prompt, I wasn't, but on the 16th of May, it was a success. So
correct, that's right. I think I saw that on social media. Congratulations.
Yeah, exactly.
Just
Thank
reading
you.
from what Daniel Espes had sent me.
Yeah, of course. And that was a few weeks ago. So today's talk is going to be about GABA and type 1 diabetes and its potential as a regenerative therapy. First, just a short outline of the presentation. So I'll begin with introducing why we in the first place became interested in exploring GABA as a potentially regenerative drug in T1D. And following that, I'll present our recently published study on the same theme, which we named Regenerate, going through the study design and outcomes in terms of safety, beta cell regeneration, and the counter-regulatory hormones. And then after that, it's open for a discussion about the future of GABA and type 1 diabetes. Are there new paths that could be or should be explored? So first, the background. GABA, or gamma aminobutyric acid, is a well-known neurotransmitter within the central nervous system. But outside of the CNS, it's found in high concentrations in both pancreatic beta cells and in immune cells. This combination has made GABA particularly interesting from a type 1 diabetes perspective, since the insulin-producing beta cells are destroyed by an immune attack. And it becomes even more intriguing when considering that GABA is synthesized from the amino acid glutamate by the enzyme GAB, glutamic acid decarboxylase, which happens to be the target of one of the major autoantibodies associated with type 1 diabetes, namely the GAD autoantibodies. And this has led to that GABA has been studied quite intensely over some years now within the context of type 1 diabetes. And there have been findings of interest in diabetes mouse models and in experimental studies using human islets. GABA is shown to stimulate beta cell growth in several ways through neogenesis, that is formation of new beta cells, proliferation, in other words, replication of surviving beta cells, and possibly also transdifferentiation through conversion of glucagon-producing alpha cells into insulin-producing beta cells. Or I said beta cells, I think, but I meant alpha cells to beta cells. Although that last thing has been debated because the findings couldn't be replicated in some later studies. But beyond the beta cells, GABA has also demonstrated immunomodulatory and particularly immunosuppressive effects strong enough to reverse diabetes in mice. This dual role in promoting beta cell regeneration and dampening the immune response is especially promising, as newly formed beta cells would otherwise risk being destroyed again by the immune system. So after these experimental studies, the key question sort of remained as to could these effects seen in the lab also be seen in humans? And in order to investigate this, we conducted a clinical trial, which was recently published in Scientific Reports. It was an investigator-initiated study in collaboration with Diamede Medical. The aim was to evaluate the safety and potential effect of oral GABA Remigen with a focus on beta cell regeneration in individuals with long-standing type 1 diabetes, which we deemed as at least 5 years of disease duration. The study was randomized into 3 different treatment arms, where arm 1 received GABA 200 milligrams daily, arm 2 received 600 milligrams of GABA, and arm 3 received 600 milligrams of GABA combined with 0.5 milligrams of alprazolam during the first 3 months, followed by GABA alone for the remaining 3 months. And the addition of alprazolam was based on preclinical findings suggesting that benzodiazepines can enhance GABA's regenerative effects on beta cells by acting as a positive allosteric modulator, or a PAM, and thereby creating a synergistic effect. To assess outcomes, 2 main methods were used. We used the hypoglycemic clamps to evaluate the hormonal counter-regulatory response to low blood glucose, and repeated mixed meal tolerance tests to stimulate insulin secretion and assess changes in C-peptide during the course of treatment. This is a busy slide, but it's meant to give you sort of a visual overview of how the visits were structured for each participant, where each column represents one study visit. The blue boxes represents the MMTTs, and the pinkish ones represent the hypoclamps. And the 3 arms were essentially similar in design, but arm 1, the first 4 participants in arm 1 were evaluated by a data safety monitoring board before arms 2 and 3 were allowed to start. So this led to a delayed start for arms 2 and 3, and as a result of that, those participants underwent an additional MMTT at baseline. Here is a flow chart over the study. I think it's quite small on your screen, but a total of 49 men with type 1 diabetes with a duration of at least 5 years were screened, of whom 35 were included in the study. 26 individuals completed the entire study, but 33 were still included in the so-called full analysis set, which primarily focused on safety. And over to the results, and starting with the safety results. Of course, you're not expected to read the entire table, but it lists all recorded adverse events, both likely and unlikely, related to the treatment. There were 2 cases of serious adverse events, 1 likely related to treatment involving elevated liver enzymes, and 1 other related to a severe event of hypoglycemia, which was unrelated to treatment. In general, most adverse events were mild and resolved spontaneously, but what sort of stood out was the elevation in liver enzymes, which we observed in 17 different occasions in 9 individuals. This led to an early treatment discontinuation in 2 participants. However, since 3 individuals in each group were affected, it doesn't appear to have been dose dependent, at least. Now, did we observe any effect on the beta cells? Unfortunately, no. Here you can see longitudinal data within each group regarding C-peptide area under the curve, with each line representing 1 individual. And the reason it sort of looks like there are a few lines, it's because they are all clustered near the bottom of the graph. So we saw no change from baseline in any of the groups, neither with the standard C-peptide assay nor with the ultrasensitive method, which we also used. In other words, we couldn't find any indication of regeneration of functional insulin-producing beta cells in any of the groups. But we also investigated indirect signs of increased beta cell mass, such as an improved glucose control, but we couldn't identify any improvement since both levels of HbA1c and continuous glucose monitoring data remained unchanged from baseline as well. So here we're looking at the CGM data concerning time below range, time in range, and time above range, which was unaltered from baseline onto end of study treatment and until follow-up in all 3 groups. In addition to that, the counter-regulatory hormonal response to hypoglycemia during the clamp studies was unaffected as well, as can be seen here in the delta values of each counter-regulatory hormone. The green colored boxes represent arm 1, pink is arm 2, and yellow is arm 3. And we looked at longitudinal differences both within groups and between groups, but we didn't find anything holding promise as a significant alteration in the counter-regulatory response. As to levels of GABA, they were measured as nadir concentration and were also unaffected from baseline until follow-up. Out of a safety perspective, this was positive since there was no accumulation of the controlled release GABA compound. But you can always speculate if we, during the study, didn't reach a high enough concentration in order to activate a possible regenerative effect of the beta cells or not. So what we can conclude is that we couldn't see any signs of beta cell regeneration in our study. Neither did we see signs of altered counter-regulatory hormonal response to hypoglycemia, but adverse events were quite common. I'd like to finish off with acknowledgements to the team behind the study. We're pretty hard to picture in one frame, so an evening picture of Uppsala will have to act as a substitute this time. Okay, so now I think that's the end of the presentation, and it's up for discussion.
Great. Thank you very much. I hope everyone was able to sort of follow. I think a couple of the slides were a little blurry. I'm not really sure why, but
Okay.
But the paper itself, you can find those figures in the paper easily, and the links are in the chat.
I'm sorry about that. I
No
can walk you
worries.
through the pictures if you want to as well. No problem.
Okay, if anyone has a specific question.
Yeah,
So yeah,
of
let's
course.
open it up to questions. And Dave Nutt, do you want to unmute yourself?
Yeah, can you hear me?
Yes.
Yeah,
Thank you,
perfectly.
Henrick. Yeah, that's so I'm a sort of GABA brain guy, as you might know.
Oh, nice.
And so this was really intriguing to me. Obviously, I knew about the enzyme being the target for the immune attack, but I hadn't realized that you thought GABA had some kind of prophylactic, or I didn't quite understand the theory. Maybe you've got a few minutes. You could just, what was the evidence before that there is a GABA receptor on the islet cell that doing
Well,
some.
what got us interested was all these preclinical studies showing that in diabetic mice models, that GABA could actually reverse diabetic mouse models to make them go from hyperglycemic state to normal glycemic state. And also when islets were isolated, we could see that adding GABA led to higher proliferation of beta cells as well.
Oh, I see. Okay. So then I suppose the implication would be maybe you need to start it when they first get the diabetes. Would that be, because maybe it's just too late. You're going to people with established diabetes, maybe
Yes.
Yeah.
So there are different aspects of this. What we tried to do was to look at the regenerative aspect, which is the neogenesis. So we wanted to see if we were able to make the patients go from 0 to something, not to restore insulin completely, but you just go from 0 to something that could protect them from things such as severe hypoglycemic events. And there has been another excellent study in the States in newly diagnosed type 1 diabetics, a pediatric study, where they also gave GABA in higher doses to these children. And that was more they were trying to look at if they could increase beta cell proliferation, because these kids obviously have remaining beta cells functioning. And then they wanted to see, can we make them proliferate? And also look at the immunomodulatory aspect, because GABA has, beyond the beta cells, GABA is also the immunomodulatory effects that's intriguing as well.
Thank you.
Our aspect was more the regenerative aspect.
I'm sorry, I don't want to hog the questions, but
sorry.
I don't understand. Why is it in the cell in the first? Do you think it's in the cell in the first? That's simply metabolic GABA, as in all cells, or do you think there's a special place for GABA in the islet cell?
There is. It seems to be in both the immune cells and in the pancreatic cells. For some reason, it is elevations in the concentration of GABA as in the brain. So that's what makes it pretty intriguing, this combination of immune cells and pancreatic beta cells. And that's what sparked interest in the first place, I think.
I did not know that. Thank you.
No, thank you, Dave.
Dave. And then Matthias van Hereth.
Yes. So it relates to the underlying mechanism. As some of you might know, and Novo Nordisk was interested in this approach maybe several years ago, and we tested it jointly at that time, maybe in slightly different settings, with Patrick Coulombat, and it resulted in a publication in Cell Metabolism, because what we called it seemed not, the animal studies just didn't seem robust. And what I mean with that is, I'm not taking myself out of the equation. We published this paper in Cell Metabolism also for our oral insulins, and this concerned my own studies. Right? So it's not like I took anything out and say, oh, the other ones have no robust stuff. Our stuff is always robust. We actually discontinued our oral insulin approaches in Novo Nordisk for that reason. Not because anything was wrong, just because for drug development, we couldn't get it most solidly to work, just to be clear. And we could not decide in that scenarios whose experiments were right or wrong. We just couldn't. You just stand there and you say, Matt did this oral insulin before, now we did it here. We get different results, and it was wobbly, at best. And it was the same for the GABA study. So based on this, I have always questions to the rationale of bringing this into the clinic, because when these things wobble, whatever you want to call it, are not robust, it doesn't mean anybody's wrong. Even means we might be able to get these things to work ultimately, but we would have to know what we are missing in general as researchers, as humankind. I still feel oral insulin would be great for type 1 diabetes, but if you ask me, what are we missing? How do we make this more robust? Other than maybe giving people 2 grams of oral insulin to have better tolerance, I have no suggestion. And 2 grams of oral insulin, in that case, will solidly kill probably 50% of the participants. So we don't know how to develop some of these things. So that's why I bring this up, because it's a general issue. And in this particular case, of course, I'm on the other side. It was not in my unit, so I didn't regenerate the data. It was a different unit. So that's my comment. Maybe a little bit critical, but yeah.
No, it's not. From my point of view, it's not critical. I'm in the curiosity level here of GABA, and reading more and more about GABA, I can't really wrap my head around it. And I think you called it wobbling, and that's exactly my feeling about GABA as well. We find this, but another group found this, and you can't really know what the mouse studies are actually conclusive of. But we thought that it was at least enough interesting to data to give it a shot. And I think we did that in a good way, but we couldn't find any results. And that's the thing with mouse studies. Mice are not men.
No, I mean, mice are not men. It's difficult. And we tried, the only way we tried to weasel our way out of this with the mice is that we at the end said, if a given entity alone or in combination cannot reverse diabetes, we don't bring it forward. I mean, you could say, if it is a preeminent example in mice, you can take the GLP-1 drug class. You can cure with semaglutide shot, you're going to cure all these NOD mice. Probably because the regenerative capacity of beta cells in NOD mice is a completely different question, is high, like in a 2-year-old human maybe, but not like an adult human. And to somehow circumvent this over-optimism that the mouse model would lend you, we just make it more stringent. But that doesn't mean we fully understand the mechanism underlying that predictive power. We don't. I agree with you on the curiosity. The curiosity remains. Like in my case for the oral insulin, because it's unnerving, we worked 10 years on oral insulin. I've written like so many papers. I often discuss this with Jade Schuyler. And then it was tested in humans, in this case also, and it couldn't be brought forward, not we, but TrialNet in that case. And it leaves you with a stale, unsatisfied feeling. And we as humans missed something where we could have overturned a few more stones that would have maybe bring some of these things to the finishing line. But that's
well. Can you guys talk a little bit? Can you guys talk a little bit about the timing? So for instance, like even this study, the GABA study, it still reinforces the importance of sort of early intervention, which really aligns with Diamed's current clinical positioning, right? So they've identified a time where their product actually works. So maybe it is all about the timing, right? Maybe the timing has to really be honed down. And then also, I think this study also challenges this assumption that the GABA pathway modulation is sufficient alone for beta cell recovery. So maybe you need 2.
Yes.
You need more than 1 medication. What do you think of that?
Yes, I think yes. If GABA is going to hold promise in the future, I think that's the only pathway forward from my point of view. I don't think that it can stand by itself. And the outcome of diabetes is complicated. And I think moreover than the timing, perhaps it's different from an individual perspective as well, that it might work in some individuals and some not. And perhaps mostly due to its immunomodulatory effects, because the immune response in the development of type 1 diabetes isn't the same in all individuals. So the immunomodulatory drugs that we give probably can't look the same to each patient as well, if that's the path that we want to go.
If you imagine, though, that just sort of a thought experiment, that type 1 diabetes is a relapsing remitting disease prior to full-fledged or full-on presentation of type 1 diabetes, then maybe the people you're getting up until that point, you really have to be cognizant of, like, are they in a relapsing state or in a remitting state when you're giving them these medications? And even when they are diagnosed, what's their status? How can you measure baseline and start and make sure what their baseline is compared to other trial participants.
And not only look at the C-peptide perhaps and antibodies, but perhaps look at the immune response in other ways with cytokines and so on.
Yeah, better biomarkers even after diagnosis.
I think so.
Matt, is your hand still up here? You still have something?
Yes, I still have something with all the comments you raised. Monica, there's a lot to unpack. I can give you, they're important questions. I can give you answers to my opinion to some of the things. So the timing.
We'll take it.
The timing, definitely. And my thought is, though, that the pathology per islet remains pretty constant, whether it's like stage 1 or stage 3 even. So that means, in principle, something that should work in prevention should also at least preserve some C-peptide as a gatekeeper, ideally, right? And then in reference to your comment, yes, in any disease, if you intervene early, it's definitely better than late because you have organ destruction, right? Just for that matter. You also said in terms of relapsing remitting, that's a different topic a bit. My opinion is, because we wrote about this with Kevin Harold a while ago, I would say the only practical conclusion it has for now, until maybe there are better markers, that could take a long time, is we have to treat or cover or try to induce tolerance or reduce immune reactivity or improve beta cell health throughout. And you saw this in the abatacept trial, the TrialNet run that only treated 1 year in stage 1. They had some effect, but they didn't reach endpoint. Probably one could surmise that if they treated the whole time till everybody reached stage 2, it would have been better. The problem that comes in with that is because you have to treat essentially healthy patients for so long, you ideally would have close to 0 side effects. So it should be an antigenic tolerance, a beta cell regeneration like we talk about here, or something that has these mechanisms that you can justify treating some individuals for 9 years and just say, look, you stay on this, going to be good for you because stage 2 will occur late. So these are the 2 comments. In terms of endotypes, I'm still on the fence. It's something we have to be vigilant about. Many times when we try to look at the responders versus non-responders or develop markers, this hasn't been too successful. And in the pathology, other than in younger patients finding more cells, which is not really necessarily another endotype, there is not much to go by. Like we don't have a TNF-mediated diabetes, and then on the other hand, we have an interferon-mediated diabetes, and on the other hand, it's only NK cells, and the other hand is only adaptive. I discussed this with an alopecia expert recently. In alopecia, you have actually innate and adaptive-driven subforms. Could be in type 1 that this is the case, but evidence not there yet. It would obviously, for the reasons brought up by you, would be very good to know. And then you need a marker and say, hey, Matt, you're developing an NK-mediated diabetes. Here we have a drug for you. Another person gets another diabetes, right? But we don't know yet, I think, in my opinion.
What's your thoughts around the microRNA markers?
The thought is early days, and my thought has always been with omics and markers and new markers. If we don't look, the only thing we learn for sure is that we learn nothing. So we should keep on looking. It's important, and technology is becoming better. We have done some things with immune AI. They're pretty good in single cells. I think if you ask, is this even because of the lack of success to find progression marker and responder markers, is this something we should close the book on? Definitely not. We are not at a stage where we can say our technologies are so fantastic that we say, despite all this, we haven't found anything. Technologies are getting better, and we have to keep on looking before we close the book, because otherwise we might preclude us from really important insights. That's my thought about that. So kudos for all of those who are looking and show that persistence, because certainly, like in any science, we don't look, we don't learn like nothing,
Yeah, I mean, I always try to bring, this is sort of my last comment, but I always try to think about this in terms of breast cancer. So when initially breast cancer treatment was sort of one and done, the big gun, and that was it. And then as people realize, or scientists realized it was so endotypal, and then drugs were developed and combinatorial treatments were developed, that's almost like a roadmap in a way, even though type 1 diabetes is not cancer, obviously. There's something to be said about getting granular on type 1 diabetes in terms of its treatment. It seems like that's starting to happen more now than ever before. So it's encouraging.
I mean, we are just slower because we can't poke around in the target organ, right? In breast cancer, in addition to the BRCA genes and the genetics, you also then have the profiling of the tumors and its receptors. And you can do this in real time in the patient who actually suffers from the disease and say, hey, look, that's your genetic makeup. This is your hormonal receptor makeup. We recommend the following therapy for you. That's easier than in type 1 diabetes. But what I fully agree with you with is we see this for other diseases, like as cancer has always led the way for these reasons, right? Mm-hmm. If we close our eyes, that would not be good. There's teachings to be learned. Even from alopecia, I always think there's teachings to be learned. I learned from Ralph Paus that they have these innate and then these adaptive forms. And I was going like, huh? And I mean, that should make us pause and think. Maybe we have not precisely the same, but these subforms in diabetes. When I always argue against endotypes, I don't argue against the principle in general. I mostly argue that we are not there yet, and we should not get overeager to subdivide diabetes in 80,000 subforms before we have clear data. But that's very different argument from not looking. We should.
No, that's true. That's true. Also, versus breast cancer, type 1 diabetes is so much smaller. So then you're really dividing your pie. It's too small for industry's liking. So.
Well, the breast cancer is a good example. Once there were solid data, they started dividing the pie. It's true that the pie also there gets smaller, and maybe type 1 pie is already smaller to start with. But of course, once you have solid data, a targeted drug development, even if it helps fewer patients, yes, it gets smaller market-wise, but efficacy also gets so much better, that maybe in some cases, if the efficacy then is so much better, some of this, it's not only market because it can be set off by a price. That has always been my thinking. And that's why we have to keep on looking and not be discouraged. I mean, even for big disease areas like obesity, who loses better weight than others? Where do you better preserve functional muscle mass? These type of questions. I think this will come in the next decades. We have to look at this subsets of type 2 diabetes, even in the big markets for sure, right?
For Stoner. Dave, did you want to comment from the neuroscience perspective?
Well, yeah, I've just suddenly realized that I don't understand something. Why doesn't the antibody attack GAD in the brain?
Well, different, right? It's GAD 65 and 67, isn't it?
Oh, I see. It's just the epitope.
Blood-brain barrier, yeah.
Well, but it can't just be the blood-brain barrier, because there are streptococcal antibodies which do attack the brain. Unless they're different. I mean, I don't know, but there are you get things like Sydenham's chorea because their antibodies attack. In fact, there's a whole new, you may not know this, but there's a whole new industry of specialist psychiatry around antibody attacks on glutamate receptors.
All right.
So which produce a syndrome like, some kind of like schizophrenia. So some antibodies clearly get in, but I don't know. It's fascinating that
how it works.
Destroys the beta cells, but you'd imagine there might be psychological effects, but I don't suppose there. I don't know, unless maybe there are. I don't know.
Yeah, interesting.
Well, you'll have to sign up for our newsletter and get more information around some of the papers that are coming out.
I would like to do that. How do I do that?
You can go to the sugarscience.org and you can sign up for our week in review. Right now, we reach 9,000 scientists and clinicians a week, and we curate all the literature that pertains to type 1 diabetes, not just from the type 1 diabetes world, but interdisciplinary as well. So maybe you'll find something interesting there.
Well, yes,
you will.
I'll look at it. Thank you.
Thank you. Thank you, Henrick, for this interesting and thought-provoking discussion. Really appreciate it. Thanks for all the work you're doing, and congratulations again on your MD PhD. That's no small feat.
Thank you so much, Monica. Thank you for having me.
Thanks again.
Have a great rest of the day.
Bye-bye.
Thank you.
Very much.
Too.
Thank you.
Bye-bye.
Thank you.
Thank you.